Welcome to Science with a Twist, a podcast for curious people who enjoy exploring how science impacts our daily lives. From technology that helps the fight against COVID-19 to solutions that help clean the water we drink, it's all thanks to science. In each episode, members of Thermo Fisher's scientific team talk to experts who are on the cutting edge of redefining how we exist. Let's get started.
Hello, and welcome to Science with a Twist, brought to you by Thermo Fisher Scientific, the world leader in serving science. My name is Candia Brown, and I lead global market development for the genetic sciences at Thermo Fisher, where we enable scientists around the world to better understand disease and how genetics influence science. For today's episode, I'm excited to be joined by Professor Luther King Abia Akebe from the University of KwaZulu-Natal. Professor Akebe conducts research in applied and environmental microbiology as part of the Antimicrobial Research Unit. Along with his students, his research helps drive a deeper understanding of how infections are passed between the environment, animals, and humans. Today, we'll discuss Professor Akebe's current research, his One Health approach to microbiology, and the gratifying experience of conducting research in a local community. Thank you for joining us today, Dr. Akebe.
Thank you.
We're looking forward to learning more about your research and how you're linking the environment with human infections through genetic testing. So to get started, can you tell us a little bit about your background and what inspired you to pursue this area of research?
Thank you very much, Candia. It's a pleasure of being on Science with a Twist today. I'll go straight into the beginning of my career or my desire to become a doctor. So everything began because I was born in the city. So I kind of grew up in an environment where you could easily go to the hospital. When you had a fever, you had a headache, you could easily rush to the hospital at any time, or you could call the doctor, and everything was fine. But when I got into secondary school, my dad decided that I should go study in my village. Now, my village was a totally opposite of the city. There were no roads, there was no electricity, even the water, we were using streams and rivers and everything. It was exciting to have a different side of life and living in another place. But what marked me specifically was the fact that the distances between the homes and the hospitals were so large. And on one day, there is this woman that was heading to the hospital who was almost giving birth. And because she was still like a long distance from the hospital, the baby was coming on the way. So we quickly rushed her to our yard. And it was in our yard with every rudimentary thing you can think about. I was so amazed at my young age. I was just about 12. And I saw my grandmom assisting this woman in giving birth. Just that moment, I realized my dad had always told me I would be a medical doctor. So I just realized at that time there is something. I quickly, at my young age, I quickly linked my city life with a village life. And I saw the need for doctors. I saw the need for people because this woman was giving birth in an environment where there was no nurse, there was no doctor, there was just a local midwife. That was my grandmom at that time. And so those are the things that drove in to me becoming a doctor, but I never became a medical doctor. When I went to the university, I decided to study microbiology. The whole intention was get into microbiology, get good knowledge, then write the medical exam. But on this fateful day, we were giving this homework and I had to do this whole presentation on environmentally transmitted infections. And boom, the lights came up and I'm like, I can't be a medical doctor because I don't have the money to go into the medical school. But if I can prevent the infections from leaving the environment into people, or if I can understand how they jump from from the environment to people, then I can still help people. So that is how I came into this field of environmental microbiology. Finally, I became a doctor, not a medical doctor, but then a doctor in microbiology. And so going into it until now, that is what has been happening. And just like we look at the current COVID in which we have gone, where there was a lot of rumors of it jumping probably from animals to the environment to humans. That is what we are doing there for now in my research group. We are looking at how these pathogenic organisms survive in the environment and what is the link between the environment, the humans, and the animals. And we do this using culture techniques, but specifically and more interestingly, we use genomic techniques because now we are able to understand the whole genome of many of these organisms and to compare them to see that, look, we are getting similar organisms from humans, animals, and environment. And we are trying to understand that link and to see how they jump from one compartment to another. So briefly, that is me. Thank you.
Well, I think that says a lot about your father's vision as well, that he saw that not just that you could treat the disease, but also that you would find your passion upstream of that to find the origin and the etiology of disease. So kudos to your dad and his vision. So now that you've given us a little bit of overview of what brought you to this point and finding your passion around the science, let's talk a little bit more about your research on the environment and its direct connection to healthcare. How does this work that you're doing help us to better understand illness and disease?
Yeah, before the One Health concept, the whole approach of fighting human diseases has been to look at humans, for example. While the humans have focused more on humans, treating the diseases, creating more antibiotics to treat human infections, doing everything possible to treat human infections, the veterinary scientists on their side have done everything to treat animals, especially with the high demand of animal protein that is going on in the world now. So when we go into animal production, for example, we realize that the high demand has driven this whole need for intensive farming. And what happens in intensive farming is that the animals are kept in groups, usually smaller space, that stresses them. And when they are stressed, it reduces their immune system and gives them chances or increases their risk of infection. So because of that, they get treated often. And also, one complex process with that is that you can't treat just the sick animals because it's too cumbersome and it's so time demanding. You have to look at which animal is sick to treat. So you can easily treat every animal prophylactically just by placing the medication in their feed, in their water, and you get to treat everyone. But what this happens is that it drives antibiotic resistance because at some point the organisms become used to that and then they can't get treated anymore. But when we look again, we'll realize that not only animal production is that massive, but at the same time, we need manure because now we are realizing that inorganic manures are a problem to our soils because of the chemicals, the heavy metals, and everything. So using animal manure becomes an alternative. But now we have treated the animals with antibiotics. They have now in their systems resistant organisms. We take the manure from that, which contains their droppings and the resistant organisms and resistant genes. We use it to fertilize our crops. When we fertilize our crops, especially in the areas where we are going to use maybe river water to irrigate or anything of such, when we fertilize our crops and there's heavy rainfall, for example, we have runoff that gets back into the river. When it gets back into the river, you now have communities that do not have access to potable water. And these communities use that same river that has runoff from the farm, that has manure from the farm that was treated with animal manure, you see. So that whole process now exposes the humans to resistant bacteria that were found in animals. And the same thing also applies with human medicine. In areas where we are, for example, where everything is sewered, like we have sewage treatment plants and everything and all the like, the waste that we produce also goes through the sewage treatment plant. True, we are safe with that. But sewage treatment plants are not designed to remove everything. So although they may remove up to a reasonable quantity of a reasonable number of bacteria, they are not designed to remove antibiotic residues, for example. They are not designed at times to remove antibiotic resistant genes. And these ones are transmitted back into the environment through the effluent. And again, we get that whole cycle that starts back. It leaves from our homes, it leaves from our hospitals, gets into the treatment plants, gets into the river. It is used to irrigate our crops, then comes back to us, goes back to the animals, and you see the whole vicious cycle continues. So that is where we now say to solve that issue, we can't just limit ourselves on humans. We can't limit ourselves on animals. We can't limit ourselves on the environment. We need to have that whole compartment those three compartments coming together, which therefore brings us that whole communication, which is the One Health concept.
Right. So you've outlined this complex ecosystem in the environment. We've also talked about, some of the behaviors or processes around how animals are developed and processed. We've also talked about how humans engage with the environment and the water to sustain their living. So now we've talked about two legs of that three-leg stool. But let's talk about the molecular aspect of it. So you've looked at the relationship and dependency on the environment and the behavior that humans are engaging in. But when it comes to the molecular biology, what are you finding and what are you utilizing? I would imagine that there's a plethora of traditional methods that are at your disposal to utilize, especially in some of these remote communities. But how are you using genetics and genomics in order to understand this complex ecosystem?
Yeah, that's true. Superficially, everything I described was like physical. The question we usually get is how can you then prove that what you have seen in the environment is linked to what you've seen in animals, is what is linked to what you've seen, is linked to what you've seen in humans. So traditional methods have always used you culture the organism, then you grow them, and then you have to. But these methods are limited by the fact that we introduce a selection bias because now we can only analyze what we can grow. But as many people or scientists and other people, it's the number of organisms that are out there outweighs the number of culture methods that we have. But now, thank goodness to genomic approaches, we don't need at times to isolate individual organisms. So if we go through metagenomic approaches, for example, we can have genomic samples from the environment, from humans, from the human microbiome or the animal microbiome. And through that approach, we can elucidate so many genes that we would otherwise miss if we were picking, let's say, an indicator organism like E. coli, for example, or we pick just Enterococcus, for example. You know, so now the genomic approaches, if we take just metagenomics, for example, gives us a broad picture of what is in that environment. Because at times, if we use shotgun metagenomics, let me take shotgun metagenomics, for example, that will give us this whole plethora of antibiotic resistant genes. Some of those resistant genes are found in organisms that we know. Some of them are found in organisms that we don't know. Some of the genes we don't even know them. We've not seen them in any human pathogen. But we realize that they are linked to resistance. At times, they are not linked to antibiotic resistance, but they are linked to metal resistance. And these metal resistant genes at times relate with antibiotic resistant genes because they are carried on the same mobile genetic elements. Now, these ones now come out when we start doing whole genome sequencing, where after we have picked up the general picture, we isolate specific organisms now and we sequence their whole genome. In that way, we are able to understand their genetic environment. We are able to understand where the genes are placed. We are able to understand the interaction between the mobile genetic elements and the resistant genes and the possibility of that gene being transferred from one organism to another. And when we do that, we now do our genomics, which allows us to relate our environmental isolates with clinical isolates and animal isolates and to see if they cluster together. At times, we do compare with isolates from our same environment and at times with isolates from different parts of the world to see if that is what has been happening. And we usually see that link coming in where we see an animal isolate clustering with a human isolate, clustering with an environmental isolate, like almost at 95 to 100% correlation. So that's how we have been using genetics to get into understanding that complex mechanism.
So you give a really compelling story around how to utilize next generation sequencing when you are dealing with a complex microbiome and you want to get a good snapshot of the flora.
Yes.
But you're also using other technologies like real-time PCR as part of your research. So how are you utilizing those two technologies complementary?
Okay. So before I get into genomics, into let's say whole genome, before I get into the whole genome, usually I kind of want to understand whether, because the presence of a gene, for example, may not necessarily mean that that gene is expressed. So if I look at antimicrobial resistance and I have pure isolates, what I want to see is I want to see if these isolates are even phenotypically resistant, like can then like place on my culture plates can they be resistant to the antibiotics of interest? Yes.
Mm-hmm.
But now for me to be sure that that is the organism that I'm using, I can't take a single organism and go straight to my whole genome sequencing because it's a little bit expensive at the moment, especially for us down in low and middle income countries. So real-time PCR comes in now to facilitate my work. Unlike previously where I will have to isolate my organism, I'll have to go through biochemical tests, I'll have to go through microscopy, I'll have to go through all those processes. Now with real-time PCR, even the conventional PCR, I will have to run my genes, I'll have to run a gene of interest, I'll have to run gels, I may have to expose myself to etidine bromide, which is cancerous, or if I have the means to use maybe other dyes, then that's fine. But that whole process becomes long. But with real-time PCR now, like the system I'm using, I don't need to go through that process. It takes me 59 minutes to confirm that this is an isolate, is E. coli, without having to run the gel, because immediately I extract my DNA. I grow my culture overnight. I extract my DNA. I put it on real-time PCR. I run it. Once I run it, I run my melt curve analysis. It tells me this is the peak. Because I've already optimized the systems, it allows me to know that this is the organism. And also, there are times when I don't feel like I'd want to go deep, depending on the students I'm working with. My master students, for example, don't need to go deep into the whole genomics and everything, because at the master's level, they are expected to understand the principles and the working of the techniques. So there we also do like checking, if we take E. coli, for example, we also check which virulence genes they have so that we can separate them into the various pathotypes. That is the ones that cause infection, enteropathogenic E. coli, enterohemorrhagic E. coli, and all those ones. Real-time PCR comes in again very handy, because what it helps me do is it helps me multiplex, that is running about 3 to 4 genes within my same tube, within the same reactions, and able to identify that this is E. coli and separates the various E. coli into the various pathotypes and getting the results within the same time. So you see, real-time PCR has really come into make life easier for me in that sense.
Absolutely. It sounds like it allows you to do quick characterization so you can develop a quick hypothesis and know which direction you want to go and really conserve your resources.
Exactly.
So with that said, you mentioned earlier about the work that you're doing in remote communities. So I wanted to pivot a little bit and talk about, we're talking about technologies that in many instances are in larger laboratories in the city with bigger infrastructure. The work that you're doing is in remote communities with limited infrastructure. So what is it like bringing these molecular methods and techniques to work directly in the community? And, when you have instances where you may have limited access to power in order to run your equipment. So how are you doing that? How are you balancing that?
That is the biggest challenge. And that is where the real challenge comes in. And now what happens most of the times is, if I am going to be working in some of the most remote communities, like when I was back in Cameroon, where we had to go to areas where you probably have to go for 2 days on foot when it's the rainy season, because the place are just inaccessible. In such cases, the works are designed such that we can do the barest minimum at the community level by isolating the organisms, and then we can preserve them to transport to the laboratory. But even within the cities, it's still not easy. Like, for example, in South Africa, we are having challenges with our water levels. And what happens with that is that we kind of get load shedding most times, so it becomes challenging to work, even though you are in the city, but because you have constant power outages, you get to spend more on generators and every other thing to run those things. So it's the major challenge this brings working in a rural community is it limits, first of all, the quality. It doesn't compromise, but it limits based on the objective. It limits a little bit the quality of work that you can do, because you want to go deeper. You have everything that you want. You know that you can get it, but it becomes a little bit expensive now because you can't run the facility. So you have to outsource. And if we take during the COVID, for example, with the power, during the COVID, load shedding was not load shedding. The lockdown was implemented in March, so we couldn't go to the labs anymore. And I only got access to my lab again in May. That was on the 27th of May of 2020. And by the time I got there, the powers had gone off. My -80 freezer was off. and fungi had grown in it. Now, this is a minus
Oh,
8
no.
into which fungi have grown, which means that fungi normally grow at temperatures of 25 degrees, which means the freezer had gone down to 0 and -8, and the fungi had everything. So
For a while.
what happened is I lost over 5 years of my microbial bank. That is the challenge. Now, imagine if this is in a city where the power outage is just intermittent. Now, you look at a rural community where the power is not just there completely. So it really presents a challenge for the microbiologists working in such an area. But it doesn't stop us from working, though. We still have to push because we need to bring back the science to the community, and we need to bring back solutions to the problems that they are facing.
So it sounds like many of your challenges around sample collection and storage, long-term storage, and ensuring that you can still preserve the integrity of those samples. So with these challenges, how's your partnership with Thermo Fisher helped you to overcome it?
Yeah, with Thermo Fisher, so far, as I said again, some of the works that I'm doing now actually revolve around the organisms that I've said and the whole isolation, identification through real-time PCR, and then going to the genomic. So with Thermo Fisher so far, I've been using a lot of their reagents, and the instrument that I currently use is the Quant Studio 5, which is the real-time platform, which has made life a little bit easy for me again, because earlier I was using a platform that had just a one temperature gradient, so I could optimize one system at a time. So I could optimize one reaction at a time. But with the instrument that I'm using now, it allows, with the FlexiPlex, it allows me to set to optimize 5 temperatures. Yes, 5 temperatures at the same time. So it's easier for me now to determine which is the optimal temperature to run from my gene of interest. So it has considerably reduced the time that I was taking to optimize my reaction.
Absolutely.
So
Yeah. You can do more complex multifactorial analysis
yes,
very, very
very
quickly.
quickly.
Yeah, that's critical. So tell me a little bit about how we can translate your research and the increased global interest in sustainability to help us understand the environmental impacts on health.
Translating, how can I put that? First of all, for me, I've always put it like translating research is usually taking that hardcore research within the laboratory and turning it into science, basic science, which my rural people for whom I'm working would understand. And this platform, first of all, like what we are doing is one of those means, because it's true we will get publications, high impact factor journals and everything, but the community, they do not read that. They don't.
Right.
So if we have more of science outreach approaches that allows the community to get access to that knowledge, then it becomes more impactful. It becomes more useful for the communities for whom we say we are working. Like one of the things I'm doing now is I'm working with one of my mentors in the US, and he had linked me with another lady, and we are trying to see how to translate this whole concept of One Health and antimicrobial research, antimicrobial resistance into cartoons. We'll translate them into cartoons so that even our kids who are watching TV, right, they will also be watching and learn from that. So any form in which such messages can go out to the community, for me, that is the best, because I feel like it's unfair for the communities where we do research not understanding what we are doing, because then
And to give
and to
back
give
and to
back,
give
yeah, it
back.
becomes some sort of selfish.
hmm
As we say in academia, we publish or we perish. We don't want to perish, so we keep on publishing. But what about the community where we collect the samples? What about the community where we see the water? What about the communities that we see using the polluted water? Because I've witnessed areas
Right.
where I know the quality of the water, and I'm looking at somebody in front of me using that water directly without any treatment for oral hygiene.
Right.
How do I
Right.
translate that to the person? For
Right.
me, it becomes
And your bridge is education.
yes,
Yeah, your bridge is how do you come back then and teach them what to look for, how to use cleaner, more sanitary techniques.
yes,
So it sounds like your bridge really is education back into the community and not just primary and secondary education. It's really about educating the entire community.
yes, yes.
Yeah. So what do you see as the future of your research?
Well, the future of my research lies on, how can I put it? I think more, just as you said about education, I am more bent towards training more people into understanding the complex environments in which we are. And when I say training more people, at times we may think that just talking about training the common person in the street or the community people. No, it entails even training the medical doctors. It entails even training the veterinary doctors. Because what I've realized with a lot of the conferences or the meetings that we are having or the workshop that I'm organizing with them is that when you speak with them, the clinicians, for example, they don't even understand the medical doctors. They don't even understand the impact of wrong prescription to their patients. The veterinary doctor, for him, he feels that just treating every animal makes it easy, but he still doesn't understand the impact of just treating every animal. What is it? Rather than investing more money on antibiotics, for example, to treat all animals, what if you invested in getting maybe more space, which is a problem, I understand, and letting the animals be more free, or to be freer, have more space to move around rather than being constricted, improving biosecurity and every other thing. So those are the things that I feel like the future of this is not ending now. In fact, the One Health concept has come to stay because that is the only way we are going to succeed if we are going to address human diseases. Especially when we look at, if we take COVID, for example, COVID was isolated from humans and from the environment in the first analysis.
Right.
So we already see that link. And we need to push that forward to make sure that it goes right from the grassroots to the top scientists and that this whole concept is fully integrated.
Well, I have a prediction, just like your father, that you'll also be teaching our young scientists as well as clinicians the value of genomics and why they need to understand and utilize these methods and techniques, not only in discovery, but also applying it into health and treating disease.
Yeah.
And I think that that's also your passion and your mission. So thank you again, Dr. Akebe, for telling us about your research and what you're working on now. And to any of our listeners on Science with a Twist, if you're interested in learning more about Dr. Akebe or any of the other health approaches in microbiology, you can visit our website for more information. So I'd like to thank our guest, Dr. Professor Akebe, for his time today and for his call to action to think of science in a more holistic way, using genomics to explore the implications between environment, humans, and animals. So until the next time, this is Science with a Twist. Thank you.
Thank you.
Thanks for tuning in to this episode of Science with a Twist. This show is brought to you by Thermo Fisher Scientific, the world leader in serving science. If you enjoyed this episode, then follow Science with a Twist wherever you get your favorite podcasts.